Hey guys, welcome. So hello guys, welcome to Ashutosh Coaching Center. Now today we are going to start chapter number 5, and the name of chapter is Principle of Inheritance and Variation. Now ये chapter काफ़ी important है for the exam के point of view से, okay? वैसे भी जेनेटिक्स आई थिंक बहुत ज़्यादा ही important होता है किसी भी एग्जामिनेशन के लिए. चलो स्टार्ट करते हैं. Have you ever wondered why an elephant give us an birth to a baby elephant, do not other animals, okay? Or why a mango seed from a mango plant and not other plant? And give they do, and of the offspring are identical parents, or they show differences in some of their characteristics. Have you ever wondered why the siblings sometimes look so similar to each other, or sometimes even so different? There are several related questions are dealt with scientifically in a branch of science, biology, known as genetics, and subject deals with the inheritance as well as the variation of the character of from the parent or offspring. Inheritance is the process by the which characterized process from parents to progeny, and it is the basis of heredity, and variation is the degree by which progeny differ from the parents. Human knew from the early as 8000 to 1000 BC, and that the one of the cause variation was hidden in the sexual reproduction, and exploit the variation that were naturally and present in the wild population of the plant and animal to selectively breed and select the organism that possessed desirable character, for example through the artificial selection and the domestication from the ancestral. इस सब का ध्यान से देख ले तो ये ज़्यादा इम्पोर्टेंट है फॉर द एग्जाम जैसे पॉइंट ऑफ व्यू से, ओके? इसका भी स्क्रीनशॉट ले ही लेते हैं. आगे देखते हैं. तो यहाँ से अब आता है हमारा पेरेंटल लॉ ऑफ इनहेरिटेंस है, बट उससे पहले कुछ और भी देख लेते हैं. वाइल्ड काउ एंड वाइल्ड काउ वी ऑल्सो we have well known the Indian breed, and example साहिवाल काउ इन पंजाब. साहिवाल काउ है जो पंजाब में रहता है. and when we must however recognize that though our ancestors knew about the inheritance and character and the variation, they had tried to little idea and the scientific basics of these phenomena. Mendel's law of inheritance क्या कहता है? It was during a midnight it was the mid-19th century that headway was made in the understanding of the inheritance. George Mendel conducted by the hybridization experiment on a grass peas for 7 years, 1856 to 1863, and proposed the laws of inheritance in living organisms. During the Mendel's investigation into a inheritance pattern, it was for the first time that the satisfactory analysis and the mathematical logical applied to the problem in biology. His experiment had a large simplicity in size, and he gave a greater credibility to the data and that he collected. Also, the confirmation of his inheritance from the experience on a successive generation of his test plant, and proved that result point to the general rules of inheritance rather than beings and unsubstantiated. समझ रहे हो? Unsubstantiated and ideas. Mendel investigate character in the grass pea garden pea plant that were manifested as two opposing traits. Example: tall or dwarf plant, yellow or green seeds. This allowed him to set up a basic framework rules for governing inheritance, which was expanded on by later scientists to account for all the universe natural observation, okay? Observation and the complexity of inheritance in them. Mendel conducted the such artificial cross-pollination experiment using several true breeding pea lines, and true breeding is the one that having undergone continuous self-pollination, and so the stable traits, inheritance, and the expression for several generations. Mendel selected 14 true breeding peas and the plant varieties, and as 14 pairs of which were similar except for one character with constructing traits, and some of the constructing traits selected were smooth or wrinkled seed, येलो एंड ग्रीन सीड्स। इनिशियल देखो, इसको याद कर लेना, ये काफ़ी इंपॉर्टेंट है। मेंडल्स ने क्या क्या यूज़ किया था उसको एक्सपेरिमेंट में। इन्फ्लेटेड फुल, इन्फ्लेटेड और constricted grid or yellow pond and tall or dwarf plant, figure 5.1। इस फिगर को भी आप देख ले ध्यान से तो ज़्यादा बेटर होगा। ओके, नाउ ये भी काफ़ी एग्जाम के पॉइंट से काम आएगा। जैसे कि सीरीज नंबर दे रखा है, स्टीम है, स्टीम हाइट जो होता है वो टॉल और ड्वार्फ होगा, या फिर समझ सकते ही हो ना। 5.2 Inheritance of one gene. Let us take example of one such hybridization experiment carried out by the Mendel's, where he exposed the tall and dwarf pea to the study and studied the inheritance of one gene, and figure 5.2 he collected the seed produced, and as a result of this cross and grew to the generate plant of the first hybrid generation. This generation is also known as filial F1 progeny or F1, and the Mendel observed that all the F1 progeny plants are tall and like its parents, none were dwarf, and he made a similar observation for the other pair of traits, and he found that F1 always resemble either one of the parents, and that the trait of the other parents was not seen in them. Mendel them self-pollinated and the tall F1 plants, and to be surprised found in that in the filial generation some of the offspring were dwarf, and the character was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf and 1 by 4th of the F2 generation F2, while the 3 by 4th of F2 and plant dwarf trait were identical to their parental and type blending. That is all offspring were that is all the offspring were either tall between height. Okay. Now similar result were obtained in the with the further other traits that he studied. Only one of the parental traits are expressed in the F1 generation, and while at the F2 stage, both traits were expressed in the proportion and 3.1. The constructing trait did not show any blending at either F1 and F2 stages or F2 stages. Okay, इसको भी आप जान ले ज़्यादा बेटर होगा, एग्जाम के पॉइंट से बहुत इंपॉर्टेंट है। नाउ based on this observation, Mendel proposed that something was being stably passed down and unchanged from the parent to offspring through the gamete over the successive generation. He called this factor now we call gene, and a gene therefore is a unit of inheritance and the content formation that is required to express particular trait in an organism. Genetic code is of for a pair construction trait are known as allele। देखो जेनेटिक होता है, एक जेनेटिक कोड जो होता है, जेनेटिक कोड जो कि एक कॉन्स्ट्रक्टिंग पेयर बना के रहता है, ठीक है ना, जिसको हम लोग एलील कहते हैं। ओके, एल दैट इज दे आर स्लाइटली different from the gene. If we use alphabetical symbol for each gene, then identical then capital letter is used to the traits, and at the F1 stage, the small alphabet for the other traits, and for example in case of the other character of height T is used to the full trot trait, then T for the dwarf, and the T and the small capital T and then small t are the allele for each other. Hence the plants of plant the pair of allele for the height would be capital T capital T or capital T or small t. Mendel proposed that in the true breeding tall and dwarf and P variety and the allelic pairs of for gene height are okay identical or homozygous. Capital T and small t respectively. Capital cavity and small t are called genotypic of the of the plant, while the descriptive terms tall and dwarf are the phenotypic. What would be phenotypic of the plant and had be genotypic small capital small t? As the Mendel found, and the phenotypic of the F1 heterozygote and capital T small t to be exactly like capital T parent is appearance. He proposed that in a pair of dissimilar factor or one dominate the others, and hence is called the dominant. Okay, हाँ dominant क्या होता है ये भी जान लो ऐसे. And the recessive and factors the other recessive. In this case T for tallness and tallness and is dominant for over T is for मतलब बोलते हैं dwarfness. That is recessive, and he observed identical behavior for all other character tall traits pairs that he studied. It is convenience and the logically and the use of capital and the lower case of an alphabetical symbol to the remember this concept of dominance and recessive. Do use capital T for tall and small d for dwarf, because you will and find to it you will find it difficult to remember whether T and D are allele of the same gene and the character nor are not. Allele can be in the similar of homozygous and capital T small t, or can be dissimilar and as in the case of heterozygous capital T small t. Okay, and the capital T small t is the heterozygous of gene controlling one character height, and it is a monohybrid, and the cross between the capital T and small t is monohybrid cross. Okay, from the observation that the recessive parental trait is expressed without any blending in the F2 generation, we infer that when the tall and dwarf plant produce gamete by the process of meiosis, and the allele of the parental pair of separate or segregate from each other, and only the allele is transmitted to a gamete. This segregation of allele is random processed, and there is a 50% of chance gamete containing either allele has been verified the result of crossing. In this way, gametes of tall capital T plant have built allele for capital T, and the gamete for the dwarf planet small t have the allele. Uh, During the fertilization of two alleles from capital T from one parent, say, and through the pollen, and the small t from the other parent, through the egg and the united produce zygote, and that have one capital T allele and small t allele. In the other words, hybrids have capital T small t, since these hybrids contain alleles with express contrasting traits, and the plants are heterozygous, and the production of gamete by the parent, the formation of zygote and F1 and F2 plant can be understood from the diagram and called Pinnet square. ओके, तो बाद में क्या हो गया, एक डायग्राम दिया गया, स्क्वायर मतलब कहते नहीं है समझने के लिए, जिसको हम लोग पिन्नेट डायग्राम के नाम से जानते हैं, एज शोन इन फिगर 5.4. ये हमारा है जिसको पिन्नेट स्क्वायर कहते हैं. ओके, डरने की बात नहीं है. as on here. It was developed by the British genetist and regarded and C. Punnett. It is a geographical representation of calculate to the probability of all possible genotypic of offspring is a genotypic cross, and the possible gametes are written of on two sides, and usually the top row and the left column. All the possible combination are represented in the box below in the square, and which generated a square output from the Punnett square and shows the parental capital tall capital T male and dwarf small t female plant, and the gametes produced by them and the F1 the F1 capital small t progeny and the F1 plant of genotypic capital T small t are self-pollinated. The symbol of male and female are used to denote the female eggs and the male pollens and of the F1 generation respectively. Genotypic is capital small t when the cell pollinated and the produced gamete of the gamete of the Cathania, capital small t and in equal proportion. When fertilization takes place and grains of genotypic T have a 50% to chance to pollinate egg and the बोलते example genotypic T as well as the of genotypic T small t also pollen grains of genotype to a 50% chances pollinating eggs of genotypic T as well as of uh, as well of genotypic T small t as a result of random fertilization. The resultant zygote can be of the genotypic capital T small t capital T small t or small t. From the Punnett square it is easily to seen that in the 1 by 4th of the random fertilization lead to capital T and 1 by 2 lead to the to small t capital T small t and 1 by 4th of to small t through the F have a genotypic capital T small t but phenotypic characters seen in tall at F2 and 3 by 2 fourth of plant as are tall where some of them are capital T while the other are capital T small t. Externally it is not possible to distinguish between the plant and with the genotypic capital T and small t, and hence the within the genotypic pair capital T small t only one character T is expressed, and hence the T is the T or tall and is said to be dominant over the other allele T and small t or dwarf character. It is thus due to the dominant and of one character over the other. All the F1 are tall all through the genotypic is capital small t, and in the F2 3 by 4th of the same or the plant are all, and through the genotypically 1 by second of are capital T small t and only 1 by 4th are capital T. This lead to a phenotypic and the ratio of 3 by 4th tall and 1 by 4th of capital T plus 1 by 2 plus 1 by 2 in small capital T small t, okay. And 1 by 4th cap small t that is a 3 ratio 1 3 is to 1 ratio and but a genotypic ratio of 1 is to 2 is to 1. The 1 by 4th and 1 by 2 is 1 by 4th is to 1 by 2 is to 1 by 4th is to ratio of capital T and ये बहुत इंपॉर्टेंट, इसको भी याद कर लीजिएगा, ओके, डरने की बात तो नहीं है, ओके. Ratio of capital T is to capital T is to small t is a mathematical condensable to the form of binomial expression ax plus by whole square that has the gamete bearing the gene capital T or small t in equal frequency of 1 by 2. The expression is expanded as a given below 1 by 2t plus, ये भी काफ़ी इंपॉर्टेंट है इसे भी जान ले, 1/2t या 1/2t प्लस होल स्क्वायर या ये जो इक्वेशन होता है ना भाई, होता क्या है, आई एम नॉट बिलीव समझो। ये इक्वेशन ज़्यादा इंपॉर्टेंट नहीं होता है, बट ये इक्वेशन जो होता है आपको मेंडल जी के बारे में ज़्यादा कुछ समझाएगा, और इसे आप अकेले में देखिए ज़्यादा बेटर होगा। तो इसे हम स्किप करते हैं, यहीं पे पूरा इवेल्युएट किया गया है और कुछ तो है नहीं। बट इसमें आपने एक चीज़ देखा होगा, तो कहीं ना कहीं ए प्लस बी का होल स्क्वायर जो होता है ए स्क्वायर प्लस बी स्क्वायर प्लस 2ab होता है। बट यहाँ पे क्या होगा, जब आप मल्टीप्लाई करोगे फर्स्ट वर्ड, जैसे आपने मल्टीप्लाई किया 1/2 का कहा ना, ए प्लस बी का होल स्क्वायर, इसका स्क्वायर होगा कहीं ना कहीं। तो ओके, जैसे 1/2 का आप स्क्वायर करते हैं, 1/2 कैपिटल टी का स्क्वायर करोगे स्मॉल टी का, तो इसमें क्या होगा, जो सॉरी गलती हो गई है, जैसे समझो, जैसे मान लो 1/2 है, यहाँ कैपिटल टी का बाहर स्क्वायर है, तो कहीं ना कहीं आप देखोगे 1/4 आएगा, और यहाँ पे जो कैपिटल टी का स्क्वायर नहीं हो जाएगा, बल्कि टी इंटू टी, यानी कि ये हो जाएगा टॉल कैरेक्टर, ये समझना होगा, बाकी सब वही है। ओके। जैसे मान लो फिर लास्ट में आएगा 2ab आता है, कोई दिक्कत फॉर्मूला में, तो 2 मल्टीप्लाई ए क्या है, मान लो 1/4 है, बी क्या है, मान लो 1/2 है, और उसके साथ कहीं ना कहीं एक चीज़ और दे रखा है ना हमारा, कैपिटल डी और स्मॉल टी। और अगर ध्यान से देखो तो फाइनल आंसर हमारा, इसको ये कट जाएगा तो आंसर आ जाएगा हमारा 2 एंड ये, ओके, तो आंसर हमारा फाइनल आ जाएगा 1/2 या 1/4 आ जाएगा इनटू कैपिटल टी स्मॉल टी। ओके, गॉट इट। चलो, Mendel self-pollinated the F2 plant and found and dwarf F2 plant continue to generate dwarf plant, and in F3 and F4 generation he concluded that genotype of the dwarf has was sorry बोलते नहीं homozygous small t. What do you think he would have got, and that had he self-pollinated a tall F2 plant? From the preceding paragraph, it is clear that through the genotypic ratio can be calculated using mathematical probability by the simply looking and at the phenotypic of a gene of the dominant trait. It is not possible to know that the genotypic composition, and that is example either whether a tall plant and from the F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, determine the genotypic of plant at F2. The Mendel crossed the tall plant from F2 with a dwarf plant, and this he called test cross. Okay, in a typical test cross, and the organism P plant here showing a dominant phenotypic and whose genotypic to be determined, and is crossed to be with a recessive parental instead the self crossing, and the progenies of such a cross can be easily analyzed to the predict the genotypic of the test organism, and figure 5.5 so the result of typical test cross over violet and colored flower W is for dominant and white for color for the small w. And using Punnett square, try to find the nature of offspring of a test cross. What the ratio would you get? And using genotypic of the cross, can you give the general definition for a let cross? test cross? Okay, let's find out. ये तो हमारा दे रखा है, इसको आप समझना, बड़े इजीली वे में दे रखा है, ज़्यादा कुछ इसमें है ही नहीं। तो अब हम आगे आ जाते हैं। Now, based on his observation on monohybrid cross, Mendel proposed two general rules to the consolidate कहते नहीं है consolidate and his understanding of inheritance in monohybrid cross. Today, these rules are called principle or laws of inheritance. Okay, and the first law or the law dominance, and the second law or segregation. Okay, इसको आप ध्यान रखेंगे, ये भी काफ़ी इंपॉर्टेंट है। दिस इज़ वेरी इंपॉर्टेंट फॉर द पॉइंट ऑफ योर एग्जामिनेशन, यू हैव टू गो 5 पॉइंट। आपको नहीं बोला था मैंने, इस ये साइड मतलब ये जो भी सिस्टम खुल गया तो उसको बोला था मैंने। ओके, यू हैव टू गो। नाउ देखते हैं 5.2.1। ओके, ये पता नहीं, बहुत लंबा चैप्टर है भाई साहब। लॉ ऑफ डोमिनेंस क्या होता है? Characters are controlled by the discrete unit called the factors, and second factors occur in pair. In a dissimilar pair of factor, one members of the pair dominate, and the dominant and the other recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and F, and the expression of both in F2. If it also explains the proportion of 3 is to 1 obtained at the F2 and the 5.212 laws of segregation. This law is based on the fact that the allele do not any blending, and the मतलब यहाँ पे क्या होता है, एल जो तो ब्लेंड नहीं करता है, डेट एंड डेट बोथ कैरेक्टर आर कवर्ड सच इन द F2 generation, thus the one of these is not seen in at the F1 stage. Thus and the parent contain two allele during the gamete formation. The factor or the allele of a pair segment from each other, that a gamete receive only one of two of the two factors. Of course, the homozygous parent produce all gametes and that are similar, while a heterozygous one produce two kinds of gamete having one allele and with equal proportion. Okay, इसका भी आप फोटो ले ले, जितना फोटो लोगे उतना ज़्यादा बेटर होगा, क्योंकि फिगर को जो होता है अकेले में समझा करो, ये तो समझ ही रहोगे। 5.2.1 इसको बोलते हैं इनकम्प्लीट डोमिनेंस। होता क्या है तो देखते हैं चलो। When experience on peas were repeated using other traits in other plant, it was found that sometimes the F1 had phenotypic did not resemble either of the two parental and was in the between the two. The inheritance of flower colored in the dog flower and the snapdragon and the बोलते नहीं एंटी राइनम sp and is a good example to the understand incomplete dominance. In a cross between two breeding red flower, red capital T capital R and true breeding, while the flower is the white flower plant small r, and the F1 capital R small r was the pink. And when F1 was pollinated, यही बहुत इंपॉर्टेंट लाइन, सारा ही इंपॉर्टेंट लाइन है इसमें। तो देखो बात ये नहीं है कि कितना इंपॉर्टेंट, बात यहाँ पे ये है कि ये सब मेंडल जी का कहा हुआ बात सब है, तो जो कि एग्जाम में बड़ी आसानी से पूछा जा सकता है। ओके, एंड व्हेन द F1 was self pollinated and the F2 मतलब बोलते हैं self pollinated and the F2 resulted in the ratio first capital T capital R red and 2 and small r pink is to 1, small r white is to मतलब white है बस इतना ही, यार कुछ नहीं है, मतलब इस तीनों का जो है 1:2:1 का है। And here the genotypic ratio were exactly as would be except in any Mendelian monohybrid cross, but the phenotypic ratio had changed and from the 3:1 dominant and recessive, and what happened was that the R was not completely dominant and over the R, and this made possible for to the distinguish capital R small r as a pink for capital R red and small r white. The explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle this question, we must understand what the genes does. Every gene, as you know by now, contain the information to express particular trait. In a diploid organism, there are copies of each genes and as a pair of allele. The two alleles need not always be identical, as in the heterozygous. One of them may be different due to the changes that is undergone, which you will be read and further on, and in the next chapter, which modified the information of that particular allele contain. And let us take example of gene that contain the information of producing enzymes, and now there are two copies of the gene of this gene and the two allelic forms, And let us assume as in more common that the normal allele produces the normal enzymes that is needed for the transformation of a substrate as कहते नहीं है theoretically the modified allele could be responsible for the production of first normal या less efficient enzymes or second सॉरी बाई फॉल्ट ये बैक हो गया just guys wait for a second now डजेंट मैटर हम यहाँ से नेट को भी ऑन कर लेते हैं कोई डरने की बात तो है नहीं ये रहा चैप्टर ऑफ 5 स्टार्ट हो चुका है बस थोड़ी सी दिक्कत आएगी उसके लिए माफ़ी चाहिए जस्ट वेट ओनली फॉर फ्यू मिनट वेट बस हो गया now it has done वेट जस्ट हो ही गया ये तुमने पढ़ लिया लॉज ऑफ सेग्रीगेशन तो ये थे हमारे सेकंड था हमारा नॉन फंक्शनल एंजाइम्स और लास्ट हमारा नो एंजाइम्स एट ऑल चलो अब देखते हैं एक एक करके सबको कहते हैं the first case the modified allele is equivalent to unmodified allele that it will produce the same phenotypic trait and that is result in the transformation of substrate as such equivalent allele pair are very common but if the allele produces a non-functional enzymes or no enzymes the phenotypic may be affected the phenotypic trait will only be depend on the functioning and the unmodified allele the unmodified functioning allele which represent the original phenotype is the phenotypic and is the dominant allele and the modified allele is the generally the recessive allele hence in the example above recessive trait and is insane due to the non-functional enzymes or because no enzymes is produced 5.2.2.2 codominance. Till now, we were discussing the cross where F1 and resemble either of the two parents, codominance, or was in between incomplete dominance. But in the case of codominance and F1 generation resemble both parents. A good example is different type of red blood cell या फिर RBC that determine ABO blood grouping in humans and ABO blood group are controlled by the gene I तो ABO जो ब्लड ग्रुप होता है उसको कौन कंट्रोल करता है तो जीन फर्स्ट ओके द प्लाज्मा ऑफ रेड ब्लड सेल has sugar polymer okay that protrudes from its surface and the kind of sugar is controlled by the gene the gene first has three layer IAIM small I the allele IA and IM produce slightly different from the sugar allele देखो ये जितना भी ये पूरा ही लाइन मेरे ख्याल से इंपॉर्टेंट होगा तुम लोग के लिए ओके इस पूरी लाइन का फोटो खींच लो ये पढ़ ही लो कोड में बहुत इंपॉर्टेंट है, काफ़ी इंटरेस्टिंग भी है। जैसे यहाँ कह रहे हैं हमारा आई ए एंड आई भी प्रोड्यूस कहता है स्लाइटली डिफरेंट फ्रॉम द सुगर वाइल आई डज नॉट प्रोड्यूस एंड sugar because humans are diploid and each possesses any two type of three genes I gene allele and the IA and IB are completely dominant and I in the other words, and when IA and small I are present only a IA expressed because does not produce any sugar. When IA and I are present in expressed and but IA and IB are present in together, they both express in on their type uh, sugar type of sugar. This is because of codominance and hence the RBC and red blood cell has both of A and B type of sugars, and since there are the three different allele, the there there are six different combination of these three alleles and that are possible, and therefore a total of six genotypic of the humans ABO blood type. How many phenotypes are possible? तो these are the possible. Okay. इसको आप इसका स्क्रीनशॉट ले लें। हमने ले भी लिया, देखा हम लोग बहुत आगे है लेने में किसी का भी भाई, स्क्रीनशॉट और कुछ नहीं। Do you realize that the example of ABO blood grouping also provides a good example of multiple allele? Here you can see that there are more than two, and that is three allele, and the government the same character since in the individual on only two alleles can be present and multiple alleles can be found, and only when the population study are made. Occasionally a single gene product may be more than one effect. For example, starch synthesis is in pea seed and is controlled by one gene. It has two allele B, capital B and small b. Starch is synthesized effectively by the capital B homozygous, and therefore large starch grain produced in the contrast small b, and the homozygous have lesser efficiency in starch synthesis and produces smaller starch grains, and after maturation of the seeds capital B seeds are round, and the small b are wrinkled. Heterozygous produce round seeds, and so B seems to be dominant allele, but the starch grain produces are intermediate size in capital B small b seeds. So if starch grain size is considered as a phenotypic types, then from this angle and the alleles show incomplete dominance. Therefore, dominance is not a autonomous feature of a gene or the product that has the information for it. It depends as much on the gene, product, and the production of particular phenotypic from this product, and as it does the particular phenotypic that we choose to examine, and in case more than one phenotypic is influenced by the sugar gene. 5.3 Inheritance of two genes. The Mendel also worked with cross P and with plant that are differed in two character as seen in the cross, and between a C plant has seed with yellow and round shaped, and had one of and had कहते नहीं है and had seed of green color uh, green color and wrinkled seed. uh, Mendel found that the seed resulting and from the crossing parent and the yellow colored round shape and hair. Here can you tell which which of the character in the pair of yellow green color and round wrinkle shaped was dominant? Thus, yellow was dominant over green and round shaped and dominant over wrinkle. These results are identical to those and that he got when he made separate monohybrid cross between yellow green and seeded plant and between the round wrinkle seed plant. Let us use genotypic symbol Y for dominant and yellow seed color, and the Y small y for recessive green colored, R for round and seed, and a small r for the wrinkled seed. And the genotypic of the parents can be written as the capital R, capital R, capital Y, and small y. And the cross between the two plants can be written down as in figure 5.7 shown. The genotypic of the green plant of the parent plant gamete, capital R, capital Y, and the small y unit of on a fertilization to produce F1 hybrid, capital R small r and capital Y small y. When the Mendel self real hybridize and the F1 plant he would that 3 by 4th of F2 plant had yellow and 1 by 4th had green, and the yellow and the green colored cigarette in a 3 is to 1 ratio. Round and wrinkled seed also cigarette in a 3 is to 1 ratio, just like a monohybrid cross. ओके, इसको भी आप नोट कर ले, बहुत अच्छा है ये. नाउ देखते हैं लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट इन अ डी हाइब्रिड क्रॉस फिगर 5.7 phenotypic and round yellow, okay, wrinkle and yellow and कहते नहीं है round green and wrinkled green appear in the ratio 9 is to 3 is to 3 is to 1, such as a ratio observed for a several pair of character mental study. The ratio of 3 is 9 is to 3 is to 3 is to 1 can be derived as a combination series of 3 yellow and 1 green with 3 round and is to 1 wrinkled. The derivation can be written as follow, and 3 is to 1 wrinkled, okay, and the 3 yellow is to 1 and is equal to 9 round yellow is to 3 and wrinkle yellow 3 is to 3 and round and green is to 1 and wrinkle green. Based upon the such observation on hybrid cross bit कहते नहीं है cross between the plants suffering in two traits, and the mental proposed in a second set of generalization that we call Mendel's law of independence assortment. The laws state that when two pairs of traits are combined in a hybrid segregation of one pair of character is independent of the other pair of character, the Punnett square can be effectively used to understand an independent segregation of the pair of allele of genes during the meiosis and the production of eggs and the pollen in the F1 capital R small r y capital R small r capital Y small y plant, and consider the segregation of one pair of gene R and capital R small and 50% of the gametes have the gene capital R and the the other 50% have small r. Beside each gamete having either capital R small r and capital R small y small r, or it should also have the allele capital capital Y or small y. And the important thing to remember here that is that segregation of 50% of small r and the 50% is small r, and the 50% of capital R it is dependent on from the other segregation of 50% of capital Y and the 50% of small y. Therefore 50% of 50% of the small r bearing the gametes and has Y, and 50% has similar as Y. Similarly 50% of R bearing the gamete Y and other 50% Y Y, and thus there are four genotypic of the gametes, four types of pollen and the types of eggs. The four types are capital R small capital R capital Y capital R small y small r capital Y small r small y, each with a frequency of 25% of 1 by or the 1 by 4th of total gamete produces. When you write down the four type of egg and the pollens on two sides of a Punnett square in the कहते नहीं it is very easy to derive the composition of zygotes that rises to F2 plant and figure 5.7 although there are 16 square how many different types of genotypic and the phenotypic are formed note down in the formula given can you using the Punnett square data and the work out the phenotype genotypic ratio at the F2 stages and the fill in the format given is the genotypic ratio also 3 is 9 is to 3 is to 3 is to 1 series number genotypic found in first and the and accepted phenotypic 5.3.2 the chromosomal theory of inheritance Mendel published his work on the inheritance and of characteristic in characters in 1865 but for several reactions it remains unrecognized till 1900 firstly communication was not easy in those days and his work could not be widely published okay secondly his concept of genes factored and in Mendel's words as a stable discrete unit of that controlled by the expression of traits and of the pair of allele which did not blend and which with each other okay was not accepted by the contemporaries as an explanation for the apparently continuous variations seen in the nature. Thirdly, the Mendel's approach using the mathematics to explain biological phenomena was totally new and the unacceptable to many of the biologists of his time. Finally, through the Mendel's work suggested factors and were discrete unit he could not provide by and the physical proof for the existence of factor or the seed they are made of. In 1900, three scientists they rise Correns and the कहते नहीं है टिस्मार्क क्या है स्मार्क independently and radio discovered Mendel's result on the inheritance of the character. Also, by that time, due to the advancement in the microscopic that were taking places, scientists were able to carefully observe cell division, and this led to discovery of structure of nucleus and that apparent to double and the divided into just before cell division. These are called chromosome. Okay, and colored bodies they are visualized by staining the in 1902 में the chromosomal movement of during the meiosis had been worked out and Walter Sutton and the Theodore Boveri noted देखो थियोडर बोवेरी एक साइंटिस्ट है बोवेरी chromosome was parallel to the behavior of gene and used to the chromosomal movement and the Mendel's law. Recall that you have studied in behavior chromosome during the mitosis and equational division and during the mitosis reductional division and meiosis reductional division the important thing to remember that are the chromosome as well as the genes and occur in pairs the two alleles of a gene pair are located on a homozygous sites on a होमोलोगस क्रोमोसोम ओके इसको भी आप ध्यान से नोट कर लीजिए काफ़ी इंपॉर्टेंट है एग्जाम के पॉइंट ऑफ व्यू से ये जितना नहीं वो तो एग्जाम पर इंपॉर्टेंट ही होंगे ना और क्या हो ही सकते हाँ अब ये देखिए 5.3 का फिगर दे रखा है ये भी काफ़ी काम आएगा इसको भी आप नोट कर लें ओके चलो देखते हैं जरा इसमें क्या है सिग्रीगेट एट अ गैमेट छोटा सा क्या पड़ता है ड्यूरिंग एना फेज ऑफ मियोसिस फर्स्ट एंड the chromosome pair can be aligned at a metaphase plate independently of each other understand this and compare chromosome of four different color and the left and the right column and the in left column possibility first and the orange and green is segregating and together but in the right and hand column possibility and in the orange chromosome segregating with the red chromosome. Okay, इसके अंदर पॉसिबिलिटी फर्स्ट है, पॉसिबिलिटी सेकंड है. पॉसिबिलिटी फर्स्ट में क्या बोल रहा है, वन लॉन्ग रेंज वन लॉन्ग रेंज एंड द शॉर्ट ग्रीन क्रोमोसोम एंड येलो एंड लॉन्ग येलो एंड शॉर्ट रेड क्रोमोसोम एट द सेम पोल. इधर क्या बोल रहा है, पॉसिबिलिटी सेकंड में क्या है, वन लॉन्ग ऑरेंज एंड शॉर्ट रेड एंड द क्रोमोसोमल लॉन्ग येलो एंड शॉर्ट ग्रीन एंड एट द पोल. ये दोनों का है, अभी बाद में दोनों को आगे इसको नोट करके आप समझ लीजिएगा, ठीक है. क्योंकि इसको पढ़ाने में बहुत ज़्यादा टाइम खींच जाएगा और हमें ये पूरा ही चैप्टर का हर एक लाइन्स कवर करना है, जिसके लिए ज़्यादा टाइम तो नहीं है, तो जितना जल्दी हो सके उसे कवर करते हैं, क्योंकि ये चैप्टर बहुत बड़ा है. चलो आगे देखते हैं, स्किप करके, लेकिन आप इसका स्क्रीनशॉट ले लीजिएगा. ओके, ये काफ़ी इंपॉर्टेंट है पॉइंट ऑफ व्यू, एग्जाम पॉइंट ऑफ व्यू से. ओके, नाउ इसका भी आप फोटो ले लीजिए इस क्यूट से कीड़े का, ड्रोसोफिला. काफ़ी स्मार्ट दिख रहा था, लेकिन बात अब ये साइंटिस्ट है कोई, द सटन बोवेरी, बहुत साइंटिस्ट आएंगे अभी. द सटन एंड बोवेरी आर्ग्यूड दैट द पेयरिंग एंड द सेग्रीगेशन ऑफ अ पेयर क्रोमोसोमल and would lead to the segregation of pair of factory, and they carried the united the knowledge of chromosomal segregation with Mendelian principle and and called it chromosomal theory of inheritance. Following the synthesis of ideas and experimental verification, the chromosomal theory of inheritance of by the Thomas Hunt. तो chromosomal theory of inheritance किसने दिया था, थॉमस हंट ने दिया था, मॉर्गन ने, and his colleges led to the discovery and the basic of variation the sexual reproduction produced, and the Morgan worked with a teeny fruit fly and Drosophila. और उसमें किस बेटा लिया था, ड्राइसोफिला को लिया गया था, एंड विच वर तो ड्राइसोफिला का यूज़ किया गया था, ठीक है, याद रखना, बहुत इंपॉर्टेंट लाइन है. फाउंड टू बी सुइटेबल एंड फॉर द such study, and they could be grown on the simply synthetic medium in the laboratory, and they complete their life cycle, okay, in about 2 weeks, and single mating could produce a large number of progeny. Flies also was a clear differentiation of the sexes, and male and female flies are easily to distinguishable, and also it and has many hereditary variation and can be seen and low power microscopic. 5.3.3, the linkage and the recombination. The Morgan carried out where several hybrid crosses into Drosophila in a study gene that were sex linked, and the cross ring were dissimilar to dihybrid, and the cross carried out by the Mendel in peas. For example, Morgan hybridizes and the yellow bodied the white-eyed female to brown bodies and red eyes married males, and the intercrossed their F1 progeny, and he observed that the two genes did not segregate and independently of each other, and F2 ratio derivated and varies significantly from the 9 is to 3 is to 3 is to 1, and the ratio unexpected when the two gametes are independent. Morgan and his group knew that the genes group are located in on the X chromosome section 5.4, and the saw quickly that when the two genes in a dihybrid cross are situated on the chromosome, the proportion partial gene combination were much higher than the non-parental type. The Morgan attribute this due to the physical association or linkage of the two genes, and the coined in the term linkage, and to describe the physical association of the genes on a chromosome, and the term recombination to the describe the generation of non-parental gene combination. And the Morgan and his group also find that even genes were grouped on the same chromosome. Some genes were very tightly linked and showed very low recombination. Figure 5.11 cross A, while while others were loosely linked showed higher recombination. Figure 5.11 cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% of recombination, while while white and the miniature wing slowed and 37.2% of recombination. He studied Alford and the Stockland and used to frequency of the recombination between the pair of same chromosome as a measure of distance between the gene and the mapped their position of the on the chromosome. Today, genetic maps are extremely used uh, s- to used as a starting point in the sequentially of the whole genomes, as was done in the case of human genome and the sequentially project described after. ये important exam के point of view से. Now, polygenic inheritance. Mendel's study mainly described those traits and have distinct alternate forms as flower color, which are either purple or white. But if you around, you will find that there are many traits and which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just tall or short people as two distinct alternative but whole range of possible height, and such traits are generally colored by three or more genes, are thus as polygenic traits. Beside the involvement of multiple genes, polygenic inheritance also take into account the influence and the environment. Human skin color is another classic example of this. In a polygenic traits, phenotypic reflect the contribution of each alleles, and that is affected the alleles is additive and to the understand is better. Let us assume that the three genes ABC colored skin colored in humans with dominance from A, B, and C responsible for the dark skin color, and the recessive forms ABC and for light skin and colored, and the genotypic with the dominant allele capital B, capital B, small capital C with a darkest skin and color, and that all the recessive alleles small b, small c, and will have the lightest skin color. As the expected, the genotypic with three dominant allele and the three recessive allele will have intermediate skin, and in this the manner number of each types of allele and in the genotypic would determine and the darkness or lightness or of the skin is an individual. It's a 5.5 pleiotropy, and we have so far seen the effect of genes on a single phenotype or traits, or there are however instances where single genes can exhibit multiple phenotypic expressions. Such a gene is called pleiotropic gene, and the underlying the mechanism and of pleiotropic is most case is the effect of a gene on metabolic pathway which contribute towards different phenotypic. As an example of this is disease phenyl ketonuria, and which occur in humans, and this disease is caused by the mutation in genes and codes for the enzymes phenylalanine and hydroxylase, and single gene mutates and the manifest itself through the phenotypic expression and the characterized by the mental and the retardation and the reductional in hair and skin pigmentation. Okay. अब हम जानेंगे सेक्स डिटर्मिनेशन क्या होता है. The mechanism of sex determination has always been a puzzle, and before the genetics and initiate clue due to the about genetics and the chromosomal mechanism of sex determination can be traced back to the some experience and carried out in insect. In fact, the cytologist logist and the observation made in number of insects and led to the development of concept of genetic and chromosome Mull and basis of sex determination. Hanking 1891 could trace the specific nuclear structure of all the through spermatogenesis and in a few insects, and it was also observed by him, and 50% of the sperm received and this structure. Okay. And the concept of genetics and the कहते नहीं है. Hanging 1891 could be traced and a specific nuclear structure for the all the spermatogenesis and in the fuse insect, and this was all it was also observed by the 50% of the sperm and the received by structure of after spermatogenesis, whereas the other 50% sperm did not receive, and Hanking gave the name of this as X body. ओके. हैंकिंग ने ही स्पर्म का नाम बाद में क्या दिया था, एक्स बडी. इसको याद रखो, एक्स एक्स स्पर्म का नाम क्या दिया था, एक्स बडी. ओके. बट ही कुडंट एक्सप्लेन सिग्निफिकेंस, बट उन्होंने इसका सिग्निफिकेंस क्लियर नहीं कर पाया. बट फर्दर इन्वेस्टिगेशन बाय द साइंटिस्ट लेड कंक्लूजन एक्स बडी हैंकिंग वाज द इफेक्ट एज अ क्रोमोसोम. इसका फ़ोटो ले लीजिए, काफ़ी अच्छा तो है. And this that is why it was given the name of X chromosome. It was also observed that in a large number of insects of mechanism of sex determination of the XO type, that is all eggs bear an additional X chromosome beside the other chromosome autosome, and the other hand some of the sperms bear the X chromosome, whereas some do not. Egg fertilized by the sperm having an X chromosome and become females, as those fertilized by the sperm do not have X chromosome become males. Do you think the number of chromosomes in the male and female equal? Due to the involvement of X chromosome in the determination of sex, and it was designed by be the sex chromosome, and the rest of chromosome were named as autosome, and the grasshopper is an example of XO type of sex discrimination, and in which the male have only one X chromosome beside the autosome, whereas female have pair of chromosome. And observation led to the investigation of number of species to understand the mechanism of sex determination, and in a number of other insect and a mammal including man's XY type of sex determination is a seen in both male and female. and okay, XY जो है वो दोनों में देखा गया है. हाँ. And female have the same number of chromosome among the male, and X chromosome is present but counted is distinctly smaller and called Y chromosome. Okay. ओके. ये भी लाइन काफ़ी इंपॉर्टेंट है एग्ज़ाम के पॉइंट ऑफ व्यू से. ये लाइन जो पूरा है ना बहुत सही, सारा ही इंपॉर्टेंट है. पहला, दूसरा, तीसरा, ये चौथा, इस चारों को ही हाईलाइट मैंने कर दिया है. Female, however, have pair of X chromosome. Both male and female have bears same number of autosome. The male have autosome plus XY, while the female has plus XX. In human beings, in Drosophila, male of one X and one Y, whereas female have pair of chromosome beside autosome. And in the above description, you have studied about two types of sex determining mechanism, XO type and the XY type. Okay, but in both case, male productive produce two different types of gametes, either with other or without X chromosome, or B some gametes with X chromosome and some with Y chromosome. Such type of X determination mechanism is designed and to be an example of male heterogamete, and in other organism example birds, a different mechanism of sex determination is observed figure 5.12, and in this case the total number of X chromosome is same in the both male and female, but two different type of gamete in the term of sex. Chromosome are produced by the female, that is female heterogamete, and in order to have a distinction with mechanism sex determination and either described earlier. The two sex determination sex chromosome of a female birds has been designed to be a Z and W chromosome, and in these organisms the female have one Z and one W chromosome, whereas male have pair of Z chromosome. Okay, and autosome. 5.6 was sex determination in humans. It has already been mentioned that the sex determining mechanism in humans and mechanism in the case of humans XY type, and out of 23 pair of chromosome present in the 22 pair of exactly same in both and the female and कहते नहीं है. These are the autosome. A pair of X chromosome are present in the female, whereas the presence of X and Y chromosome are determined in of the male and characteristic during spermatogenesis among the males, and the type of gametes are produced, and 50% of the total sperm producer carry with the X chromosome, and the rest 50% has Y chromosome beside autosomes. Female, however, produce only one type of ovum with the X chromosome, and this is individual probability of fertilization of the ovum, and with the sperm carrying either X and Y chromosome. In a case of use ovum fertilization with a sperm carrying X chromosome, the zygote develop into a female XX, and the fertilization of ovum and Y chromosome carrying sperm and mm, result into a male offspring, and thus evidence that is genetic makeup of the sperm and determine the sex of the child. And it is also evident that in each pregnancy and there is always 50% of probability of either male or female child. The unfortunate that in our society women are blamed for giving birth to female children and have been ostracized and ill treated because of this female notion. Like this. इसको भी आप फोटो खींच ले, ज़्यादा बेटर होगा. अच्छा सा नहीं आया, सॉरी, गड़बड़ा गया, ये थोड़ा सा गड़बड़ा गया. अब ठीक आया है. ओके. Sex determination in honeybee. Sex determination in the honeybee is based on the number of sets of chromosome an individual receive, and an offspring formed from the union of a sperm and an egg developed as a female queen and worker, and a unfertilized egg as a male drawn by the means and parthenogenesis. And this means that a male and have half number of chromosome the that of a female, and a female are diploid having 32 chromosome, and are diploid and having 16 chromosome. Okay, this is called the haploid sex determination and system, and has special characteristic feature such as male produce sperm by the mitosis, and they do not have father, thus cannot have son, but have grandfather and have and and can have grandson. How is the sex determination mechanism is different from in the birds, and is sperm or the egg responsible for the sex of the chicken? Let's check out. इसका भी फोटो ले लेते हैं, बहुत इंपॉर्टेंट है ये सब. म्यूटेशन का केस आता है. अब जानेंगे म्यूटेशन क्या होता है. अभी 4 पेज है, नहीं 4 पेज अभी और है, कोई दिक्कत नहीं है, बहुत ज़्यादा है. but कोई दिक्कत नहीं है. Mutation is the phenomena which result in the alteration of the genetic DNA sequence and consequently result in the change in the genotypic and the phenotypic of an organism. In addition to recombination and mutation is another phenomena that lead to the variation to the in DNA. As you learn in chapter 6, on one DNA helix is continuously from one end to other each chromatid, and in highly supercoiled from, and therefore loss and deletion or gain insertion and duplication and for genetic of a segment of DNA result in the alteration in the chromosome, and since the gene are known as to be located in chromosome, alternation in chromosome result in the abnormalities or aberration, and the chromosomal aberration are commonly used observed in a cancer cell. In addition to above, the mutation also arise due to the change in the single base pair of DNA, and this is known as the mutation. And a classical example of such a mutation is sickle anemia cell amina. Okay, and deletion and insertion of DNA of pair of DNA and causes frame shift mutation of chapter 6 point see chapter 6 chapter 6. Okay, and the mechanism of mutation is beyond the scope of this discussion and at a level. However, there are many chemical and physical factors that induce mutation, and these are the referred to as mutagens, and UV radiation caused a mutation in the ऑर्गेनिज्म. इसमें निकलता क्या है तो म्यूटेशन. ओके, और यूवी रेडिएशन जो होता है मतलब कि जो म्यूटेजन कहते हैं कि म्यूटेजन होता है, इट इज़ अ म्यूटेजन. और यूवी जो ज़्यादा म्यूटेशन म्यूटेजन का काम करता है. हाँ, तो जेनेटिक डिसऑर्डर क्या होता है. 5.8.1 जो होता है पेडिग्री एनालिसिस. द आइडिया दैट डिसऑर्डर आर इनहेरिटेड दैट एंड हैज़ बीन प्रिवेलिंग इन द ह्यूमन सोसाइटी सिंस लॉन्ग, एंड दिस वाज बेस्ड ऑन द heritability of certain characteristic feature of in families. After rediscovery of Mendel's work on a practice analysis inheritance pattern of traits in humans beings began, since it is the evidence that the control cross that can be performed in the plant or some other organism are not possible in case of human beings, and study of the family history about the inheritance of particular traits provides any alternative. Such analysis of traits in a several generation of a family is called pedigree analysis. Okay, and in the pedigree analysis, the inheritance of a particular traits is represented in the family over tree over the generation. In humans, genetics pedigree study provide a strong tool which utilize the stress and inheritance of a specific traits. Abnormality or diseases, some of the important standard symbol used in a pedigree analysis and have been shown in figure 5.1.3 and 4.13. And as you have studied in chapter, each every feature in organism is controlled by one or other gene located on the DNA present. Okay, now in the chromosome, DNA is the carrier of genetic information, and it is hence transmitted from one generation to other without exchange, without any change or the alteration. However, the change or the alteration do takes occasionally, and such an alternation or the change in the genetics material is referred to as mutation. And the number of disorder in human beings have been found to be association and with the inheritance of changed or the altered gene or the chromosome. The Mendelian disorder. Broadly, the genetic disorder may be grouped into two categories: Mendelian disorder and the chromosomal disorder. Mendelian disorder are mainly determined by the alternation or the mutation in the single gene, and the disorder are transmitted in the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance such Mendelian disorder can be traced in a family by the pedigree analysis. Most common and the prevalent and the Mendelian disorder are hemophilia and the cystic fibrosis and the sickle cell anemia and the color blindness and the phenylop. मतलब ये सारा एग्जाम्पल बेटर है, ये सारा एग्जाम्पल बेस्ट है, सबको नोट कर लो. एंड मेंडेलियन डिस्कवर्ड मे बी डोमिनेंट एंड और रिसेसिव, एंड बाय द पेडिग्री एनालिसिस वन कैन बी easily understand whether a trait question is dominant or recessive. Similarly, the trait may be also linked to the sex chromosome in as hemophilia, and it is evidence that the X linked and the recessive trait shows transmission from the carrier female to male progeny, and representative pedigree is shown in figure 5.1.3. And dominant and recessive traits. Discuss with your teacher and design the pedigree for the character linked in to both autosome and sex chromosome. हम्म इसका भी लग रहा है ओके इसका भी फोटो ले ली जाए ज़्यादा बेटर होगा हाँ नेक्स्ट इज कलर ब्लाइंडनेस व्हाट इज कलर ब्लाइंडनेस इट इज़ अ सेक्स ब्लाइंड रिसेसिव डिसऑर्डर ड्यू टू द डिफेक्ट इन द आइदर और ग्रीन और आई मतलब ये भी काफ़ी इंपॉर्टेंट है कलर ब्लाइंडनेस क्या होता है इसके बारे में भी जान लेना ठीक है कलर ब्लाइंडनेस होता क्या है पहली बात तो अच्छे से जानो अच्छे से it is a sex linked ratio recessive and the disorder due to the defect in the either red or green cone of eye resulting in the failure discriminate between the red and green color the defect due to the mutation in certain genes and the present in the X chromosome and and it occurs in about 8% of the male and the only about 0.4% of the female. This is because the genes that lead to red green color and the blindness are the X are on the X chromosome. Male have only the X chromosome and female have two. And the son of woman who carries the gene has 50% chances of being colorblind, and the mother is not herself colorblind because the gene is recessive. That that mean that its effective is suppressed and by the matching dominant and normal gene. A daughter will not normally be colorblind and unless mother is carrier and her father is colorblind. Hemophilia क्या होता है? This is a sex linked, ये सारे इंपॉर्टेंट है सेक्स linked recessive and diseases and which shows transmission and from the unaffected carrier female and to some male progeny and has been widely studied this in this disease and a single protein that is a part of cascade and of proteins and involved in clotting of bloods and affected due to this is an in an affected individual and simple cut will result in non-stop bleeding and the heterozygous female carrier for hemophilia and male transmit the disease and the son the possibility of a female becoming hemophilia and extreme rare because mother of such female has to list carrier and the father should be hemophilic and unviable in the large later stage of life and the family pedigree of Queen Victoria shows a number of hemophilic descents as she was a carrier of disease sickle anemia and this is a autosome linkage recessive that can transmitted from the parent to offspring and when the both partner are carrier of the genes or heterozygous this disease controlled by the single pair of HB or HBS or out of three possible genotypics only the homozygous individual and HB5 HBS and showed the disease and the phenotypic heterozygous HBA HBS and individual appear apparently unaffected but they are the carrier of disease and as there is a 50% of probability of transmission of the mutation gene into the progeny and thus the exhibition a sickle cell trait and the defect is caused by the substitution and the glutamic acid ओके इसका भी फोटो ले लेते हैं नाउ आगे बढ़ते हैं ये चैप्टर अभी तक सबसे लंबा चैप्टर लग रहा है ये तो द ग्लू बाय द वैलिन एंड वॉल एंड एट द सिक्स पोजिशन एंड द ऑफ द बेटा ग्लोबिन चेन ऑफ द हीमोग्लोबिन एंड द सिचुएशन एंड द सब्स्टिट्यूशन ऑफ द अमाइनो एसिड इन द ग्लोबिन एंड द प्रोटीन रिजल्ट ड्यू टू द सिंगल बेस सब कॉन्स्टिट्यूएंट एंड कहते हैं substituent at the sixth codon of the beta globin and gene from the gag and to jug the mutant hemoglobin molecules undergoes polymerization and under low oxygen tension and causing the change in the shape of RBC from the bioconcave and disc to elongated sickle like structure next ये भी काफ़ी अच्छा है phenyl ketonuria and the inborn error of metabolism also inherited autosomal recessive traits and the affected the individual lack an enzymes and the convert the amino acid and the phenyl align to into the triosine as a result of phenylalanine and is as accumulated the and the converted into a phenyl pyruvic acid and the other derivatives. Accumulation of these brains is a result in a mental retardation, and these are also excreted through the urine because of its poor absorption by kidney. Thalassemia. Okay, this is also autosome linked recessive blood and disease transmitted from the parent to the offspring when the both partner are unaffected carrier of the genes heterozygous, and the could be could be due to the either mutation or deletion which ultimately result in the reduced rate of synthesis of one globin chain alpha and beta chain makeup and the hemoglobin. This is caused the formation of abnormal hemoglobin and the molecules resulting into anemia, which is characteristic of the diseases. Thalassemia can be classified according to the which chain is hemoglobin and the molecules is affected, and alpha thalassemia production of a globin chain is affected, while B thalassemia and alpha production of B globin chain is affected by the alpha thalassemia is controlled by alpha A thalassemia is controlled by the closely linked by the HbAI HbA2 and on chromosome 16 chromosome of each parent and is observed due to the mutation or the deletion of uh, one or more of the four gene, and the more gene affected the less alpha and the globin molecules and the white alpha and the white beta thalassemia and is controlled by the single gene HBB on chromosome 11 of each parent occur due to the mutation of one or both gene. Thalassemia differ from the sickle cell anemia in the former is a quantitative problems for synthesizing to the few globin molecules, while the there later is a qualitative problems of synthesize and incorrectly functioning globin. 5.8.3. Okay, इसका भी फोटो ले लो भाई, बहुत काम आने वाला है सब। क्रोमोसोमल डिसऑर्डर क्या होता है? क्रोमोसोमल डिसऑर्डर ऑन द अदर हैंड आर कॉज ड्यू टू द एब्सेंट ऑफ एक्सेस और द एबनॉर्मल अरेंजमेंट ऑफ वन और मोर क्रोमोसोम, द फेलर ऑफ segregation of chromatid during the cell division or called cell result in the gain or lost chromosome and is called aneuploidy. Okay, for example, down syndrome result in the Down syndrome is result in the excreta copy of chromosome 21 similarity Turner syndrome and due to the loss of X chromosome in the human female and failure cytokinesis and after telophase and of cell division। ये बहुत इंपॉर्टेंट चैप्टर भाई, ये मेरे दिल को छू गया ये लाइन जो था। इस लाइन को समझ जाओ भाई, मज़े आ जाएंगे। बहुत तगड़ा लाइन था भाई सब। अब ये बार वॉल्यूम क्यों क्लिक हो जाता है? कभी वॉल्यूम क्लिक होता है, एक से क्लिक होना होता ही नहीं है। ओके, अब बाहर द फेलर ऑफ साइटोकाइनेसिस आफ्टर द टेलोफेज एंड द स्टेजेस ऑफ सेल डिवीजन एंड रिजल्ट इन एन increase in a whole set of chromosome in an organism, and this phenomena is known as polyploidy, and this condition is often seen in the plant, and the total number of chromosome in a normal human cell is 46 या 26 pairs. Out of 22 pairs are autosome and of one of chromosome are sex chromosome, and sometimes through the rarely either an additional copy of chromosome may be included be an individual and ओके इसका भी फोटो जरूरी है। Individual may lack of any one pair of chromosome. This situation are known as triosime and the monozyme or a chromosome of a chromosome respectively. Such a ये वर्ड काफ़ी इंपॉर्टेंट है, ट्रायोसाइम। and monozyme भी important है. Okay, and Turner syndrome, Klinefelter and syndrome are common example for the chromosome disorder. Down syndrome क्या होता है? This, देखो डाउन सिंड्रोम टर्नर सिंड्रोम तीनों ही इंपॉर्टेंट है, ये तीनों ही इंपॉर्टेंट है बहुत ज़्यादा एग्जाम के पॉइंट ऑफ व्यू से. नहीं पढ़ रहा है भाई साहब. अब ठीक है, डाउन सिंड्रोम क्या होता है, इसके बाद कितना बचेगा, लास्ट ही है हमारा. इसका भी फोटो ले लो, ज़्यादा बेटर होगा. डाउन सिंड्रोम. The cause of genetic disorder is the presence of an additional copy of the chromosome 21 trisomy, and the disorder was discovered by the Langdown and 1866, and affected by the individual is short stature with a small round and the head, and the furrowed tongue and the partially open mouth, and the palm is abroad with characteristic palm, and the creates physical psychomotors and the mental development of retardation. Nile filter syndrome, and this genetics disorder also caused due to the presence of an additional copy of X and chromosome, resulting into a karyotypes of 47XXY, and such an individual has overall masculine development, and however the feminine development of breast and the gyno कहते नहीं है ये gynocost cosme gynecomastia is also expressed figure. Such individual are sterile. टर्नर सिंड्रोम क्या होता है? Such disorder is caused due to the absence of one of the X chromosome, that is 45 with XO, and such female are sterile as ovary are rudimentary besides over the feature including. Okay, now lack of other secondary sexual character, figure 7 point. ओके, अब नाव पढ़ लेते हैं इस लाइन को. इस लाइन को भी बहुत इंपॉर्टेंट ध्यान से पढ़िएगा. Genetic is a branch of biology which deals with the principle of inheritance and its practices and progeny resembling with the parents, and in morphologically and the physiologically feature has attached to attention of any biologist. And the Mendel was first to study the phenomena systematically, while the study pattern of inheritance in the pea plant of contrasting characteristic, and Mendel proposed the principle of inheritance, which are the today referred as a Mendel's law of inheritance, and he proposed that the factor and later named genes regulating the character found be in the pair of alleles, and he observed that the expression of the character in the offspring follow a definite pattern, and in the different first generation F1 and second generation F2 and so on. Some characteristics are dominant over others, and the dominant characters are expressed when the factors are in heterozygous condition, law of dominance, and recessive character are only expressed in the homozygous condition, and the character never blend in heterozygous condition, and recessive character that was not expressed in the heterozygous condition may be expressed again when it becomes homozygous. Hence the character segregates, and while the formation of gamete and laws of segregation, not all the characters show the true dominance, and some characters show incomplete and some show codominance. When Mendel studied inheritance of two characters together, it was found that the factor independently assort or combined two permutations, and the combination law and the law of dominance independence assortment, and the different combination of gamete and theoretically represented in a square tabular form known as the Punnett square, and the factor now of known as a gene or a chromosome regulating chromosome are called genotypic, and the physical expression of chromosome of character are called phenotypic. After knowing that the genes are located in on the chromosome and good correlation was drawn between the Mendel's law and segregation and the assortment of the chromosome during the meiosis, and the Mendel's law were extended in the form of chromosomal theory of inheritance. Later, it was found that the Mendel's law independent assortment does not hold, and the true for the gene were located on the same chromosome, and these genes are were called as the linkage gene. Okay. And the closely located gene assorted together, and the distantly located gene due to the recombination assorted, and the dependent linkage map therefore the correspond to arrangement and of genes on chromosome. Many genes were linked to the sexes or also called the sex-linked chromosome, and the sexes male and female were found to have a set of chromosomes which were common, and other set which was different. The chromosome which were different in two sexes were named sex chromosome, and the remaining sex set was named as autosome. And in humans, normal females has 22 pairs of autosome and pair of X and X, and male has 22 pair of autosome and pair of sex chromosome X and Y. And in चिकन में जो होता है एक्स वाई जेड होता है और जेड डब्ल्यू होता है, ठीक है. एंड म्यूटेशन इज डिफाइंड एज द चेंज इन इससे याद रहा मुर्गी का. Mutation is defined as a change in genetic point, and mutation is a change of base pair in the DNA. Sickle anemia caused due to the change of one of the base in the gene coding for the beta chain of hemoglobin, and the inheritable mutation can be studied by the generating a pedigree of a family. Some mutation involves changes in the whole set of chromosome polypeptide, or change in a set of chromosome number aneuploid, and the help in the understanding the mutation basis of genetic disorder. And the Down syndrome is due to the trisomy of the chromosome 21, where there is an extra copy of chromosome, and consequently the total number of chromosome become 47. And the Turner syndrome, one X and X chromosome and is missing, and sex chromosome is as XO, and in cal कहते नहीं है Klinefelter syndromes, and the condition is double XY, and this can be studied in analysis karyotypes. Okay, so guys, thanks for the watching video till the end, and you are a part of journey, so please like, share and more subscribe, and जितना हो सके उतना ज़्यादा शेयर करो. बाय, see you in next video.